guys. So this is our notes for our next unit, IS4, which is genetics or inheritance of traits. And we're going to be spending some time working through some problems in this video. I'm going to forewarn you, it is a long video. I am going to ask you to stop and do some practice problems. Please make sure you actually do that when I ask you to, and that you don't just fill in the answers as I move on in the video with the answers. It's not going to help you. You really need to actually spend the time to practice the problems and make sure you're understanding how we get to where we're getting. If you do not understand it, you need to bring it to class and ask questions. If you don't ask questions, I'm assuming you're getting it. So please make sure that you do the notes, that you do them as early in the week as you can so that you can ask questions, like on Thursday and Friday in particular. It would be great days to ask questions. And I just want to make sure that we're having enough time to do all that stuff that we need to do, okay? Alright, so with that, let's go ahead and get started on our notes. I've got the, whatchamacallit here, the notes packet here. Please make sure you print it out and write on it or type directly on it. I'm actually surprised at the number of you who are typing straight on here. I'm very impressed. That would drive me nuts, but good for you. Also, we've got the notes part here, so I'm going to go ahead and get started on that. Alright. And I've printed out my own packet, and I've got my document camera here. I finally got that set up so that we can go through some of these practice problems instead of doing it on the board, because I realized the sound quality was not that great when I did that. So I'm all prepared to hopefully do way better. My fingers are crossed. Okay. Alright, genetics. Here we go. The genetics is also called heredity, and that's when you have the passing of traits from parents to offspring. That's kind of the whole thing. We had this in our video last week. The study of heredity is genetics. There's a cute little comic strip here. It says, your tests show that your condition is hereditary. But the guy is like, yay, great, good, send my bill to my parents, because, you know, you don't want to pay your own bill if you can blame your parents for it. It's way better. Alright. Gregor Mendel. He is the Austrian monk who was known as the father of genetics. He's got an interesting history. If you ever get a chance to look him up, usually I spend some time talking about it, and we won't get into that today. But yeah, he's got a very interesting history. Alright, so his experiments. Remember, this is from our learning targets sheet that you were going to be able to explain his experiments. What he did is he studied pea plants. These were awesome because they were widely available and they grew really fast. And that's important because you can have multiple generations in a very short amount of time, so you can observe changes that are occurring from generation to generation over that relatively short span of time. If you were studying humans, our lifespan is much longer, and our gestation is longer, everything is longer, and it's not easy to see changes over generations because you're not alive for more than 3, maybe 4 if you're lucky generations. The other thing, so they were readily available, they were easy to grow, so that they were not difficult. Some plants are rather difficult. I personally have a little bit of trouble with tomatoes. I love to grow tomatoes, but I have a horrendous time growing them without getting blossom end rot and other things. So these guys were easy, and as I mentioned, they grew fast. Alright, so after doing his experiments, he made some conclusions, which were that each parent has 2 separate factors for a particular trait. This is again from the video. We had factors in parentheses. We now call those genes. Okay? I think we missed a slide. We did. That was slide 6. I'm so sorry. Go back to slide 5. Fertilization is the joining of male and female reproductive cells. A hybrid is an offspring of crosses of parents with different traits. Okay? So if you have a, like they have lots of plant hybrids of plums and apricots. If you mix those together, you get a pluot. That is a hybrid. Okay? Now we can draw his conclusions about genes. He looked at seed shape, seed color, the seed coat, the pod shape, the pod color, the flower position, and the plant height when he did all of his studies and his experiments, and he compared all of those things to figure out what was going on, or try to figure out what was going on, although he could not put that in the terms that we do today. Slide 7. Genes and alleles. Genes are sequences of DNA that code for a specific trait. Alleles are the alternative form of a gene. So you might have a gene for, let's say, yellow seed color, yellow seed color, but you could have two alleles. You could have a yellow and a green allele. So you would have the same trait being seed color, but you would have the two different alleles. Also, another example would be eye color, and your eye color could be, maybe you have brown eyes, but maybe you have an allele for blue and an allele for brown. And we'll talk about why they're brown instead of blue in a minute. Okay, which thumb do you put on top? If you do this, this is actually genetic. It's kind of interesting that you can see recessive, and I don't remember which one is the dominant way to do it, but I think if you're right-handed, your right thumb will go on top, and if you're left-handed, your left thumb will go on top. There is also a really interesting one. I'm going to show it to you. If you put your finger, your middle finger to your thumb, if you bend your wrist up, if you have this little part pop out right here, that is actually a recessive trait, and it's a tendon that we don't need anymore, and some people have it, some people don't. I believe when I did this, both of my kids have this trait, so it's kind of funny. You can now find out about that weird tendon in your arm. Alright, some terms that you will probably need to know. Okay, not probably, you will need to know. First is P generation. This is your parental P generation, okay? That's the parents, okay? The F1 generation is your first generation of offspring, so that's the kids, right? F2 is the second generation of offspring. That would be, if you wanted to look at human structure, that would be the grandkids. So this would be grandparents, parents, kids, parents, kids, grandkids, if you want to look at it either way, okay? So make sure you get that. Alright, so some more terms that we need to know. Traits. Purebreds have a pure pair of alleles. So each of these letters here is an allele, okay? This is your gene. It has two alleles. So we have a pure pair, has both of them either capitalized or lowercase. So big T, little t, big B, little b. And that can stand for any sort of things. You'll see what I'm talking about in a few minutes. A hybrid, though, is your mixed pair, and that's where you have one big letter and one little letter. So we have big T, big B, big G, little g, and big S, little s. And that's kind of important when we get to our Punnett squares, that you understand your pure pair, your homozygous, or your heterozygous mixed pair ones. And we're going to talk throwing those vocab terms in there for you. All right. A dominant trait is always going to be expressed when it is present, meaning when it's seen. So that is our capital letter. We always write dominant traits as our capital letter. That's the way we know that it's dominant, okay? So there's some examples. So up here, of the mixed ones, the first letter is your capital letter. That's your dominant trait, okay? The recessive trait, though, can be expressed, but only when it's in a pure recessive, so a purebred recessive one. So it's hidden in hybrids because the dominant trait is expressed. So the recessive trait is expressed here, and the recessive trait is expressed here. It's written as our lowercase letter, and you have to have two lowercases together in order to get that trait expressed. Okay? All right, Mendel's principles of dominance. Some alleles are dominant, but others are recessive. He is the first one to figure that out, or to put words to it, okay? A dominant allele will cover up or mask a recessive allele, okay? Like you can have the gene for blue eyes, but you have brown eyes. That's because blue eyes is a recessive trait, if anybody did not know that. And in order to have blue eyes, you have to have two recessive blue eye genes come together, okay? So, let's do some practice on this, shall we? And I'm going to switch over to a document camera real quick so we can practice this. All right, on here, try to make it as straight as possible. I'm not always the best with this. I apologize in advance. Okay, so we have several traits. So we have T's, G's, and B's. Our big T is tall. Our little t is short. Our big G is going to be green. Our little g is going to be yellow. And our big B will be black. And our little b will represent white. Okay? So if we were to compare big T, dominant is capitalized. So remember, dominant equals capital letter. And recessive equals lowercase. Okay? Just remember that. So big T is going to give you tall. Big T, little t is also going to give you tall because it's got that dominant trait. But little t, here's where our pure two recessive genes come together and we get a short trait expressed. Okay? How about big G, little g? What are we going to have for that one? We will have green. Little g, though, is two recessives. So two recessives is going to give us yellow. So that's our recessive trait being expressed. And our big G can only give big G's, so it can only give green. So we have green. Okay? Last but not least, we have our black versus white. So two dominant blacks will get you black. Two recessive whites, little b's, are going to get us white. Okay? And a heterozygous, a mixed hybrid one, will give us black. Okay? So hopefully that's something that you guys can practice. And I'm going to switch out of this. Trying to do this quickly. I'm getting better. Give me patience. There we go. And this is it, our answers. Let's see how we did, if it loads. Alright, so our big T's is we have tall, tall, and short. And we have green, yellow, green, and we have white, black. Oops, that's in a different order. Ha! That's funny. Good to know. I will fix that. Well, I probably won't this time. So our big B is in the middle. So we have black, and then we have little b white, and then we have big B, little b black. This will actually tell me who's copying it versus who's actually going through the video with me. This is my check, my cheat check. Just kidding. It's not. All right. Mendel's laws of heredity. He has two laws, the law of segregation and the law of independent assortment. So, law of segregation is when two alleles separate when gametes are formed, which is during meiosis, and we'll talk about that in a few minutes. Okay, so they're going to, when you have your gametes formed, they're not going to stay together. They're going to move into different cells. The law of independent assortment is you have alleles of different genes separate independently of one another during gamete formation, which is during meiosis. That means your genes are not stuck and linked together. So hair color isn't tied to foot length. Okay, it's going to be a separate thing. Okay. The other thing to put on here is that gametes are your sex cells, and they contain one copy of each chromosome. That's why you have two gametes coming together, and when two gametes come together, then you have your full set of chromosomes. Okay? So we're going to do some more practice. All right. What does this say? Height and fur color independently and randomly mix, such as you can have tall versus short with red or black fur. So our possible combinations, and this is silly, I put the answer right on there, are you could have, sorry. Okay, so we take our height first and we can put those out. So we have tall versus short, okay? And then we take the fur. Well, tall can have red fur and tall can also have black fur. It doesn't matter. You can have either. They're not linked together. Similarly, short can have red fur, but short can also have black fur. Okay, so that means you could have any combination. So that's a lot of opportunity for some variation amongst the individuals of the population, which is important. Sorry. Riley, quiet. Okay, I got them a little quieter. Let's go on. All right, so we have Mendel's law of segregation. So we have two parents on here. We have the dark blue and the light blue chromosomes. The chromosomes have doubled up before we get to meiosis, and that is important because your single chromosome here will wind up with doubling. So instead of just 2, you have your 4, because remember, we haven't gotten there yet. Just kidding. There's 4 chromosomes. So you have 2 chromosomes. They double up and they make 4. Each of those goes into its own cell as a pair. And then in meiosis 2, each of those gets split up so that our gametes have half the number of chromosomes, okay? So you have one parent giving you your dark blue gene, and then you have another parent who is giving you your light blue gene. Now this one is a dominant, the dark blue is dominant, and the light blue is recessive. So when you get down here, you have a hybrid pair, okay? Does that make sense, hopefully? Hopefully. If you need a little bit more explanation, this is page 172 in your textbook, figure 11-5, and you can read through the little caption underneath there. Okay. All right. Concepts of heredity. An offspring will inherit 2 alleles for a trait, 1 allele from each parent. The combination may be either homozygous or heterozygous. And now we're going to talk about those 2 terms, because this is what we will use pretty much exclusively. We won't use pure pair or hybrid. We will pretty much, well, hybrid you'll see, and you'll have to know that hybrid is heterozygous. But homozygous means the 2 alleles are the same. You either have 2 dominant or 2 recessive. So 2 capital letters means you have a dominant homozygous, and that the examples here are big B, and big T, big T. A homozygous recessive will have 2 little letters. And that, if you remember on our slide before, we had homozygous dominant, so we had 2 big letters, and we had homozygous recessive, we had 2 little letters. Okay? All right. Then, for heterozygous, that is when you have 2 different alleles, 1 capital and 1 lowercase. So we have big T, little t, big B, little b would also be another example of that. Okay? All right, concepts of heredity. Genotypes are the set of alleles an individual inherits for a trait. So that would be your big R, your big W, your big T, little f, those things. Okay? Your phenotype, though, and hopefully you remember this from last week's vocab, is the physical expression of a trait. So let's say you have freckles, that might be little f to get freckles. Okay? Let's say you have blue eyes, that might be little b. Maybe you have dimples. Maybe that is big D, or big D, little d, okay? So any combination can give you, so actually more than one combination can give you the same phenotype depending on whether it's a dominant or recessive trait. Hopefully that makes sense. All right, Mendel and probability. I'm not copying this down, we're just going to read through this real quick. Whenever Mendel performed a cross with pea plants, he carefully categorized and counted the offspring. For example, whenever he crossed 2 plants that were hybrid for stem height, big T, little t, or heterozygous, about 3/4 of the resulting plants were tall and about 1/4 were short. Okay? That's important because this is starting to get us talking about our proportions and our probabilities. So if you do one tall that's if you do 2 heterozygous talls, then you get this offspring combination, and it always works out to this percentage. 75% will be tall, with 25% being short. And we'll talk about that in a few minutes. Mendel realized that the principles of probability could be used to explain the results of his genetic crosses. So probability is the likelihood, and this part you do copy down, the likelihood or chance that something will or will not occur. It is expressed in fractions, ratios, and percentages, and we will be working with multiple varieties of both. Actually, we will do all 3, so you'll be able to be very fluent in doing that. Okay? All right, we're going to practice some coin flipping here in a second. If we have one coin flipping, what is the probability of having heads or tails show up? So the probability of having heads, you have a 50-50 chance. We say it that way all the time, right? So heads would be 50% and tails would be 50%. And you could write that as either 1/2, you could write it as 0.5, or 50%. Okay? 2 coins flipping. So, 2 coins flipping. What's the probability of getting 2 heads? And then heads and tails, or heads and tails, and tails. Okay? So this one, I need to double check this. I think I'm going to have you do this one. Okay. Yeah. Okay, so we're going to switch to the other camera for a second. No! And we'll get to this right here. All right, so if we have 2 coins flipping, the probability of getting heads, as we found out on the last slide, is 1/2. The probability of the second coin having heads is also 1/2. In order to find out what the probability of getting 2 heads is, you have to multiply those two together. So 1/2 times 1/2 gets you 1/4. Okay, so you have a 25% chance of getting both of them heads. Now, what is the probability of getting one coin having heads or tails, and the other coin having, so one having heads, one having tails, one having tails, and one having heads. So having a mixed pair. So we already know the probability of having heads is 1/2. And we know the probability of having tails is also 1/2. Okay? And remember from this problem here, 1/2 times 1/2 gets you 1/4. So we have a quarter possibility, or probability, 25% chance of getting heads or tails on the first coin. Same on this side. Tails is 1/2, heads is 1/2. If you multiply those two together, you get 1/4. Now, what's the probability that you will have 1 heads and 1 tails? That turns out to be 1/4 plus 1/4, which is 1/2. Okay? So you have a 50% chance of having a hybrid in this case. All right, almost done. Tails and times 2 tails. The odds of getting 2 tails, you have the same probability of heads or tails is 1/2 times the other one, which is also 1/2, which gives us the probability of 1/4 because we multiply those two together. If you add this up, 1/4 plus 1/2 plus 1/4, you get 100%. You get 4/4, 4/4, right? Because those are the only combinations you have. So it makes sense that it's that way, hopefully, to you guys, and I'm hoping. Okay, this one is not the easiest, I understand that, and hopefully we can work through some more problems this week on it. Okay? What's the probability of having a baby girl? How would you write that? Hopefully you wrote 1/2 or 50% chance, and it's the same probability of having a boy. Okay? All right. So let's move back over to the slideshow. As it loads very slowly. Which is pretty typical. Alright, so there's our math again. Alright, Punnett squares now is a little bit, hopefully you guys have done some of this. I'm hoping, fingers crossed. Alright, Punnett squares are cool because they show all possible outcomes of a genetic cross. And those are your gamete combinations. Remember, or you don't know. So there's some rules to this. The first thing is that you're going to always use place the capital letter first on your Punnett square. And then you're always going to move gametes from each parent. So you're going to move the top ones down, so each of the boxes below get that allele. Okay, and then the second parent across. So parent 1 will go on the top, parent 2 will go on the side. Don't know why that's the way we do it, but that's just customary, okay? All right. When you have these, this is a single trait one is going to be a monohybrid cross. It's a cross with one set of traits. So this could be your heterozygous one of big G, little g. You have 2 possible gametes from each parent. So I would add that. In fact, we should add that from each parent. Let's do that right now. 2 possible gametes from each parent. Okay, that's important. And we're going to italicize that because I really want you to remember that. Okay. Which gives you then 4 possible trait combinations. Okay. I just realized we skipped the practice on there. Come on. Slide 20. Oh, we've got to do some practice problems. Okay, so this is slide 25. I apologize, guys. Sorry I didn't keep you updated with all that. And then I would definitely... I'm so having trouble tonight. Sorry. Put this down. Let's go back to the visualizer. So on here, I would definitely write, like, if you shade 1 box equals 1/4 or 25%. Okay? I would shade that one in. And then let's go back to this part here. We had our rules. Hopefully you wrote those down. And let's do some practice problems together. All right. So remember, we are going to place the capital letter first and move gametes from each parent. And then the parent 1 goes here, and parent 2 goes here, and the parent 1 gametes go down the row, and these ones go across the row. Okay? Hopefully that arrows make sense. It'll make sense when we start doing actual letters here. Okay. All right, Punnett square practice. Cross a homozygous black fur male rat with a white fur female rat. Okay, so we have 2 alleles. We have big B is black and little b is white. So we know that this is a homozygous black fur male. So we know homozygous and black fur is big B's. So 2 big B's for homozygous. And white, if it's a white female, it has to be little b's. Little b, okay? So this is parent 1 times parent 2. Take your parent 1 and bring the alleles down over here, big B, and bring parent 2 over to here, little b. And we customarily always put the capital letter first. So I always start at the top and work my way down, and if I know I have a capital letter, I just scooch it all the way over to the side, same here, so that I have room for a second letter after it. Okay? Then I take my little b's and I bring those across to both, because each box needs to have 2 alleles. Okay? Each box must have 2 alleles. Okay? Can't miss that. Don't forget that. Okay? All right, so that's the first one. Let's work on the next one. All right. My camera, there we go. Okay, we're going to cross a rabbit that is homozygous for black fur. Okay, so we again have big B, little b. So homozygous for black means that it's big B, with a rabbit that is homozygous for white, which is another little b, just like our rat one. Okay, here's where we have our P1 cross. Now this one is, we haven't talked about this part yet. This is exactly what we did on the previous page, but I did not put P1 here. This is your P1 cross. Okay, so your P1 cross is just what the alleles are. So we have our homozygous black fur rabbit, which we got the alleles right here, big B, times our homozygous white fur, little b, okay? So then you have to put, the first thing we do is put it on our Punnett square. So go big B, parent 1 goes across the top, little b goes on the side, okay? Bring your big B's down. and your little b's over. There we go. All right, so now we have some new stuff over here that we're going to deal with, okay? This is our F1, and this is, if you remember from the beginning of the notes, P1 is your parent generation, and F1 is your offspring, and F2 was the offspring of the offspring, which we aren't getting into. We will get into that in a minute. We're going to do F2 next. Okay, so phenotypes. What is our fur color going to be for all of these offspring? Well, all of them have a big B, right? So, and big B is dominant, which means they all have to have black fur. Okay, and what's our ratio of phenotypes? Well, they're all black fur, so we have to say it's 4:0 or 100%. Okay. Let's look at our genotypes. In this case, because we have two homozygous dominant and recessive, you have all heterozygous offspring. So we only have one genotype, which is our big B, little b. And our ratio, again, is the same. It's 100%. So it's 4:0 or 100%. Okay? Hopefully that makes sense. So now we're going to take, this should actually say F2. Oh no, that's our F1 cross. So now we're going to take one F1 rabbit and cross it with another F1 rabbit. So we've got our genotypes here, right? We calculated those. So we have one F1 rabbit times one F1 rabbit. Okay? Both of our parents are there. Now take parent 1, put that one on the top. Take parent 2, put that one on the side. Now we have capital letters on both the side and the top, so we have to make sure we follow the rules. We bring our parent 1 down. Big B's go first, over to be the first letter. Little B's are going to be, they're little, so they're going to be second, so I'm going to just scoochy that one over to the side, so I have space in case I have a big B coming in, which I know I do because I can see right here. So now I'm going to bring over my other big B here, okay, and my little b is going to go here. Now we have an interesting offspring set. Our ratios are not 100% anymore. So this time, what phenotypes do we have? What's this one going to be? With two big B's, it's going to be black. With one big B and one little b, this one's going to be black. With one big B and one little b, this one's going to be black. But we have two little b's here, so this is homozygous recessive. This one's going to be white. So we have black and white. Okay? And our phenotype ratio, we have 1, 2, 3 black, 2, 1 white. Okay? Now our genotypes, this ratio is going to be a little different. We're going to have 3 different numbers in it because we have 3 different genotypes represented. Always start, so our genotypes always start with homozygous dominant. So genotype, we always do homozygous dominant to heterozygous to homozygous recessive. Okay? So in this case, we have big B, because that's our homozygous dominant, to our heterozygous, which is our big B, little b, to our homozygous recessive, which is little b, little b. Now we can just put numbers to this. We have the ratio going. What numbers do we have? Well, we have one big B, so we just put that down. We have 2 big B, little b's, and we have 1 little b, little b. Okay? So that would be that. Okay? And we finished slide 25 already, so I'm going to just show you that again. So that was the good practice. This one is something that you will have a lot of practice problems on. Please make sure you understand it, and if you have questions, you need to ask. I will be putting these in the openers straight away, so you'll be getting them right away. Okay? All right, I think I'm going to stop here for today. This one's a hefty one. I think we're going to do, I'll do another part one. I think I'm going to assign both part 1 and part 2, but I think I'm going to stop this one right here. Okay?